The overall goal of this technique is to effectively capture viable circulating tumor cells, or CTCs, from whole blood. These CTCs can be placed into culture or be used for downstream analyses, which require non-fixed samples.
This method can help answer key questions in the field of metastasis, but can also be used to follow disease progression, response to therapy, and evaluate risk of recurrence. The advantage of this technique is the ability to perform label-free capture of viable CTCs from any solid tumor while remaining independent of biomarker expression.
We first had the idea of releasing CTCs from the filter because we wanted to do downstream RNA analyses and culture of CTCs instead of culturing them in situ on the filter.
To begin the experiment, weigh enough PIPAAM to prepare a 10% weight per volume solution in a 15 milliliter tube containing butanol. Vortex the tube until the solution is clear. Next, cut plastic microscope slides into roughly 12 mm by 12 mm squares with a pair of scissors. Then, using a sharp pair of straight-edge scissors, cut a slot pore microfilter wafer into 8 mm by 8 mm squares and place these pieces directly onto the plastic squares. Using a polyamide film tape, secure the cut filters onto the plastic squares, only on the edge and the corner of the filter, so that at least 7 mm by 7 mm filter area is not covered by the tape. Place the microfilter that is secured on the plastic square onto the vacuum chuck of the spin coater. Program the spin coater to the following recipe. Then,